NIH and NDS. At the NINDS, involved in neuromuscular research for a long time. And then we have Francesco Muntoni.
Well, I've also been working for a rather long time as a pediatric neuromuscular physician. I work at the moment at the Institute of Child Health at Great Ormond Street Hospital in London, UK.
And then on the sponsor side, we have Serge Braun from Genethon.
Hello, I'm Serge Braun, CSO of AFM Teleton and acting as head of neuromuscular strategy for Genethon.
And Beth.
Hi, hi everybody. I'm Beth Beluccio. I'm the global clinical lead for rare neurological disorders at Pfizer, and I'm a neurologist by training, and I'm been honored to be with this group here working on these problems together.
And Teji.
Hi, my name is Teji Singh at Sarepta Clinical Development and an intensivist by training.
And then we should have join us shortly, Kyle Morris of Solid Biosciences. Oh.
I'm here, Carsten. Sorry, technical issues. Hi, so Kyle Morris is CSO at Solid Biosciences, and also very I'm happy to be working with this great group of sponsors and clinicians to figure things out.
And then on the right, you can see our expert panel from different institutions, different backgrounds, cardiologists, immunologists, Duchenne experts, inflammation experts that are been very generous, donating their time and brains to drilling down into this problem further. Francesco.
So I think we have Carsten and the group have prepared a number of orientation slides, and I will start with the concept that you are all very well aware that the microdystrophin, that is the transgene delivered via AAV vector, is not exactly the same size as the dystrophin gene or protein that we have, just because there is no space in AAV to have everything that a normal dystrophin has. Despite this, as is mentioned, we are optimistic that the part that the dystrophin that is contained in this microdystrophin will still be functional. Next slide. And I will describe just the first part here of the slide, indicating that the dystrophin gene has 79 exons, and I can probably venture to, yes, maybe you can see my mouse. And there are 79 exons, and each of these exons produce a portion of or encodes for a portion of the protein. And this is what we all have in our muscle if we do not have Duchenne. And forward, Carsten, is over to you.
So as Francesco was mentioning, if we want to deliver a therapeutic construct, something that can take the place of dystrophin, it would be, of course, ideal to do the entire length of the protein with the two business ends that you can see in orange and a yellow-green, but everything in between as well, because nature has come up with this molecule the way it is, because it makes sense and is functional for muscle. However, because of the constraints that we have in AAV gene therapy, one has to construct versions of the dystrophin that are much, much smaller to fit into the delivery vehicle of AAV, and those are called microdystrophins. And so what you can see here is three versions of the microdystrophin construct that are in clinical trials at the moment. And what they all have in common is they have the crucial business ends. So whatever is in color is included in the microdystrophin, and what is left blank here is not included in the microdystrophin. And you can see the business ends here on this part and this part are all preserved and all common. That allows the dystrophin to form the linkage with the dystrophin protein complex and the internal parts of the muscle cell to really provide that mechanistic and mechanical link. However, a lot of the parts in the middle have been gutted out, and and really deleted, if you will, or not included to fit the size constraints. And that's guided by nature by observing patients with very mild forms of a dystrophin disorder that are missing, that have lost the internal parts of the protein and still have really well-preserved muscle function, telling us that if one has to get rid of anything in this protein, it would be these middle parts. And so that results in these three constructs that maintain the business ends, have variable parts of the middle parts preserved, so they differ in that way, so similar at the ends, but differing in the middle, and then resulting into these kind of proteins that have the business ends here in orange and green, but in the middle parts have various combinations of elements that are somewhat different between the three constructs, or four. But what's also important to recognize here is that there are differences in the way these constructs are driven in muscle and how they're packaged to get to muscle. And so the package to get to muscle is the capsid. So that's the AAV subtype, or we call it serotype, that's used to deliver the microdystrophin to the muscle. So that's our envelope, if you will, our delivery tool. And you can see all four sponsors have different versions of it. They're all AAVs, they're all closely related, but they're different in their capsid. So you can see Sarepta has the one that's called RH74, Genethon 8, and then Solid and Pfizer and AAV9. Also different between these constructs and AAV constructs in the clinic are the drivers of the transgene. This is called promoter. That's the element that allows the cell to read off the transgene and make it into protein in the cell. So those are the promoter elements are all, you can see here in different letters, MHCK7, SPC512, CK8, a muscle-specific promoter. They're all elements that are known to efficiently drive in muscle, but they're different in the molecular structure. So you can see, and that's important in our discussion, that the test articles, if you will, the tested AAVs are different in their capsid, in their envelopes, in their promoters driving the transgene. And there are comparable but different in the transgene structure. So the most important commonality is the same business ends at each end. So what happened? There was a small but distinct subset of patients in the clinical trials that appeared to have an immune reaction against the delivered microdystrophin proteins, against the transgene, and we'll describe that in a little better detail in the coming slide. But it was across different trials. So again, with different capsids, different promoters, different microdystrophin versions that still developed a common reaction against this microdystrophin that was delivered. And it resulted in inflammation of the muscle, myositis, and in some cases also inflammation of the heart, called myocarditis. So in total, this was observed in 5 patients across 3 different clinical trials. You can see the age range here from 7 to 9. And there's a range of different doses that you, from these doses are not exactly comparable between trials because the titrating methods are somewhat different, but you can see it is over a wide range of different doses given. And so the conclusion from that is that there is no relationship to the specific agent, so the specific capsid, the specific promoter, was a specific microdystrophin. And yet there was something in common to these 5 subjects in all 3 clinical trials that we have to understand, and that'll be coming up in the next few slides. Francesco is going to describe what happened clinically.
Yeah, so I think and I suppose one of the reasons why with Carsten and all companies agreed to speak is that it was clear that this may well be a problem unrelated to any particular like specific microdystrophin news, but at the same time was something that all the companies have in common. But what was in common was the fact that this patient had a striking. Can you see my mouse?
Yeah, no,
Yeah. Okay. Yeah.
no.
Okay.
Yeah. I can't see it.
There was a striking similar event, and I think this is important to realize. The time course of this event was between 24 and 42 days after dosing, and this is important because that is at the time when the promoter, as Carsten suggested, will start to drive significant expression of the protein dystrophin. The virus is there from day one, the viral vector, but dystrophin takes time to be produced. What happened was that these people developed significant muscle weakness, but also with quite an unusual pattern of distribution. In particular, voice and swallowing muscle became affected. And as you are aware, these are not common things we see in people with Duchenne muscular dystrophy, but was also a common thread between these different young people. Heart involvement by at least lab testing was observed also 1 week later in 3 of these patients, and this followed the skeletal muscle involvement. Importantly, this line says that there were no problems with kidney, liver, platelets, or complement. And I think for those who understand or have been exposed to AV adverse event, this indicates that something different from what we had experienced before was noticed. And multiple treatments were required. Some of these individuals required to be ventilated. Cardiac medication were required. And in a variable way, depending on the site, the subject, different immunosuppression regime were used. But I think importantly, all these young people improve after this quite scaring, quite frankly, event. Next slide.
So we had to understand what was really happening that's common to all of these patients, and that is common to all agents that were in clinic. And so the hypothesis is that this immune attack is directed against the region of the microdystrophins that is present in all the microdystrophins, and that the patient immune system did not recognize as self because that region is missing from the patient's protein repertoire because of the underlying genetic mutation. So let's see what that means. So again, this is a cartoon that we showed before. And again, we pointing this out because it shows the region that are common to all the microdystrophins that are in clinical use at the moment. There had to be something in common between them because it's not agent specific. It's not transgene specific or compound specific or AAV specific. The deletions, the underlying mutations that the patients with the events had are very informative. So what you see here is a cartoon of where the deletion, so the lack of genetic material was in the patients who experienced these adverse events versus patients here that did not. And what you can see here, the patients with these adverse events all had deletions of genetic material that overlaps with the protein parts, the genetic material that's now delivered with the different microdystrophin transgenes, as opposed to patients who have loss of genetic material that occurs in region of the dystrophin gene that is not represented in the microdystrophin transgenes. None of these patients experienced anything like these adverse events. In fact, they had no myositis and no myocarditis, none of them. So it really all comes down to this region of the protein that is delivered by the transgenes and is deleted in the genetic background of the patients.
So I think that this summarized now not in a visual way, but with in writing what Carsten has just indicated. So it looks as there was something in, it looks as there was something common, and this was the type of deletion and that part of the dystrophin protein that was missing in this patient. And therefore, the overarching hypothesis is that there was an immune reaction against the part of the microdystrophin that the individual people with this particular type of deletions did not recognize. They'd never seen that before because of the underlying mutation. And in a way, that concept that we recognize in other conditions, but it was a novel concept in the Duchenne field, is the possibility that these people having this particular complication may have a kind of rejection, not very different from what happens after organ transplantation.
So this is a little, we apologize for the simplicity, but it's a little primer on what the immune system learns when the body develops. So every immune system undergoes a learning process by learning to tolerate all the proteins that belong to its own body. And so this is helped by a particular group of immune cells that are recognizing all normal proteins in the body and regulate the police of the body to not attack them. So they said, well, these proteins are citizens of the body. You will not be attacked. And these are the regulatory T cells that basically establish this repertoire of immune tolerance. I suppose when something comes into the body that is not part of the citizenship of the body or the normal protein repertoire of the body, in which case the regulatory T cells will stand back and the cytotoxic, the attack T cell will come forward and attack the foreign protein. That could be a bacteria, that could be viruses, but it could also be an organ that is transplanted into the body in organ transplantation. So I have an example here of a kidney transplant. So when a kidney is transplanted from a foreign donor into someone who needs the kidney, the immune system will first of all not recognize it as part of the body or the surfaces of the different cells in the kidney, and will want to attack it, which in organ transplantation, of course, is subdued by medications that subdue the immune system and try to teach the immune system tolerance to the organ transplant. So in a way, this is happening here as well on a single molecule level. You remember the parts of the dystrophin protein that are delivered by the transgene and that are not known yet to the body as a protein because that part of instruction of the protein is missing due to the genetic mutation. So these patients who experienced the adverse events had no chance to tolerize against that part of the dystrophin body protein because it was never there. So when it now comes delivered with the transgene, there is a part of that transgene that the T cells will not recognize. And then the cytotoxic T cells step forward and say, well, I don't know what this is, I better attack it. And since it's expressed by the promoters in heart and in muscle, it will be recognizable in these two organs, in muscle and in heart, and therefore they may get attacked by the immune system as foreign, resulting in what we see as myositis or myocarditis. So that's the current hypothesis behind it. It's a rejection of a foreign, perceived as foreign protein that's delivered with the transgene due to the very specific nature of the genetic mutations that that caused immune naivete against this this part of the transgene. So what are the risk mitigation strategies going forward from what we know thus far? So we clearly identify genetic situations that put patients at risk of having this adverse events because they're immune naive against a part of the transgene. And the safest way forward at the moment is to exclude patients with genotype that may be at risk for this adverse event. We also need to institute careful monitoring of our patients, of course, for signs of this happening, muscle weakness, heart issues, in particular around that critical time when the dystrophin transgene is expressed, and inform and prepare study doctors for the way to recognize it and treat it if it occurs. We need to also define this more precisely. What exactly it confers this risk? Can we narrow this down even better? What could the adverse events mean for the long-term expression of the dystrophin transgene? Is it a one-time attack and then everything is okay? Or does it carry a longer problem in terms of the durability of the transgene effect, which means it really is a risk beyond the severe, of course, occurrence of the myositis right after administration? The additional risk factors that allow us to stratify patients that are particular risk for this and need particular care. And then, of course, are there proactive, just taking our cues from organ transplantation, are there proactive as well as reactive treatments to deal with the immune system, minimize the immune response, and foster tolerance against this transgene so that it will be accepted by the body in the longer term?
So I think that the Carsten mentioned that I pushed some of the companies. In reality, I think firstly, I tell you, I became aware that there may be a problem. I threading a careful balance between having privilege information, legal obligation to sponsor, and ethical obligation to patient. I consulted with Carsten without sharing if like details that will take me into trouble, but sharing enough information to really have some also advice from Carsten, with whom we have a long-standing collaboration and respect in terms of what would be useful. And then I, after psychotherapy with Carsten, I prepared a detailed letter without having too much information, but having sufficient information indicating to all the sponsors that it was likely that each of the program could experience this particular event. And the sponsor, I think, credit to the sponsors, they accepted the fact that there was a likely risk, and they have agreed to form this consortium. As you are aware, there are not many of this consortium where companies are happy to discuss in a pre-competitive space findings, and especially now discussing exactly what this adverse event has been achieved and is continuing, because we are gathering additional knowledge and information. I certainly learn a lot, and hopefully everybody has, by sharing this information. But I think it was also clear that while there are clever people in this group, it's also clear that it was very useful to bring in additional expertise, and that is the decision to host the group that Carsten acknowledged at the beginning, that was that allowed to take forward. So I think that in a way, this is quite an interesting and important point, that not only, if you like, we are trying to address this particular aspect for people with Duchenne muscular dystrophy, but I think it's an important point that for the sponsors and investigators, if there are things that are not proprietary to an individual condition, nobody gains if another sponsor makes mistakes in an area that potentially will affect everybody. So I think this is quite an important step, and again, I'm grateful to Carsten and to the sponsor having accepted to share this information so that we better understand and try to reduce risks. And this is called experimental medicine. And the next part, I think, is yours. Yeah.
Yeah, just a very final point. It actually goes beyond just the DMD trials that we're discussing. This is a phenomenon that's to be reckoned with in all of gene therapy, because you can envision gene therapy really as a single molecule transplantation. And you have to be aware that the immune system may or may not like that. And so the rejection of transgenes is something that is to be addressed in all of gene therapy. That's good news in a way, because the entire field is really forced to address this issue, and that'll be helpful for everyone here in this room to come to solutions more quickly, because it's beyond the sponsor, beyond the agent, beyond the actual trial, beyond the disease. It's really a thing that gene therapy needs to face at the next frontier effectively. And the tools are there. So I'm very hopeful and very positive that this will be possible in a safe and rational way. Which brings us now to, I'm going to just go back to this slide here, just to have that here as our background slide so that we all can see and be reminded of where this occurred and why this occurred. So what we'll do now is we'll go into some of the questions that were raised in writing to us beforehand, and that may address some of the questions you are having in the room, but feel free to type your questions also in the chat as we go so that we can address them as well. And so we'll go back and forth, Francesco and I, between the questions that were asked. And so the first question really is addressed by this diagram again. Are there patients with deletions that are not developing an immune response? And more importantly, which I have not addressed yet, are there patients with deletions in that particular critical region that didn't develop an immune response? And so the answer is nuanced. There are some patients in that critical region who have deletions who are in this at-risk genotype that at least clinically didn't develop an immune response that mounted a clinical event with the myositis and myocarditis. So it's not entirely black and white. You're at highest risk when you have the deletions in that critical region, but there are some patients who somehow escape at least the big problem, the big brunt of it clinically. And of course, these patients are also important to us to understand how did they become more tolerant to the deletion in that region. So that's a very good question. That's ongoing work that we need to address.
And I think that the next question is on whether all companies exclude the same patient, or if there is a difference between companies, and if so, why? I don't know whether colleagues from the company want to take this, or whether you want me to give a, if you like, I tell you a high level first. And I think this is quite an important aspect. And we know what we know, we don't know what we don't know. And this is, 8 months ago, nobody would have ever thought about this. So I think we are gathering information as we are. And I think that there is a large overlap between deletions that are considered to be safer than others. I think that it is with the information that is available today, and it is possible that this information may compress or expand in months to come. There is no specific reason to think that this is a bigger problem than what it is, but I think it is slightly difficult to give you an exon by exon and a nucleotide by nucleotide answer that is evidence-based. I think that we are gathering, and the colleagues in the companies are gathering as much information. I can tell you is nobody's interest for a patient, for people with Duchenne to experience this adverse event. And and I do not know whether the colleagues from any of the company want to add, but I just want to, if like, it's important in experimental medicine to share. Lack of knowledge is important. We may not always have the answer to every single question, and I think is why this is called experimental medicine.
And Francesco, maybe I can speak a little bit to the approaches that different companies are taking in their ongoing trials, and other of us on the call may want to speak up about it too. But I think at Pfizer, the approach that we took was because we obviously have the most access to our own data. Our external data monitoring committee looked at all of the patients that we have treated so far, both in our phase 1 study and in our phase 3 study. And they tried to look at some of the patterns that are being described here, which patients with deletions had the SAEs and which patients had mutations that didn't have SAEs, and trying to sort that out. And also looking at some of the immune responses that we've been collecting on our trials. And based on just our own data set, we took an approach of trying to exclude patients that we thought might be at risk, just to be sort of conservative, even if in the end it turns out they're really not at risk. We wanted to be conservative and sort of cover all of our bases at this point until we can do additional research. And I think that other companies have really done the same. So because we all have slightly different complete data sets that we're looking at, I think there's not 100% overlap in the steps that we've taken going forward. But at least from our point of view, that's because we had the greatest understanding of our own data set, and our external data monitoring committee could really dive into that. Whereas this effort here that's collaborative and very important, they didn't have access to everybody else's data. I think going forward, as we continue to collaborate and continue to learn more and more, we're likely to come to a consensus and be able to to proceed forward in a way that's more and more unified as we gain more information. But for now, I think each of us have taken the approach of making changes to our studies that align with our own data, and that's why there's slight differences across the different trials.
Can I just say something, Beth, while you because in a way I think is an important aspect what you just said. I think that my sense, but I would like to know whether that is the case, because in a way I think I can see that for families, for advocacy group, there will be some slight difference between one particular, although actually they are almost overlapping entirely, but there are some subtle difference. I would like perhaps the people who are listening to have the comfort that one of the advantages of having this working group now is that should one of the company develop something that is similar to this, that may make patients, so say, I think that there are other regions of the gene that at the moment are not being considered to be of any relevance, but should one of the company experience a very similar event, now there is an alarm bell that instead of being me needing to connect with Carsten and so on, I think that there will be a different thread. And therefore, a big advantage is that, there is no alarm bell, and that things should occur again with this mitigating factor. But however, should that be, there is now a working group that will probably take an hour to share information. Before, it took maybe a week to align everybody, and it was very quick for industry standard, quite honestly. So I think that is, I just wanted to, and I can see that Teji and Beth and other colleagues are nodding. So in a way, it may not be so crucial if one particular study has this particular exon or not. The question is, should there be a problem? Will we learn from it? Will the company learn from it? There is no alarm bell. This should be a problem, but there is now a system to deal with that.
I mean, I would echo what Beth said, and honestly, I think I'm really, really grateful for Professor Muntoni and Professor Bonnemann to actually get us together. I think one of the biggest advantages here is we have a much faster communication across companies, sharing of information. But as Beth said, there will be some differences by the construct. Each of our companies have a slightly different construct, a different database. I pick on our own particular company. What we've done is excluded ones that we have, where we had the event is when we expanded, but we know if we went back to our first few studies that they were relatively safe. I also want to point out that there's a two-pronged thing here, at least I'll pick on Sarepta. We want to keep advancing to even get an approval of a therapy. We still have to prove gene therapy does work for DMD boys. So while we keep advancing those studies, we're going to be learning more about these at-risk patients. What makes them at risk? Is it the mutation? Is it the mutation plus patient characteristics? Because again, it's not all identical. So I think there will be slight differences, but a lot of overlap, and I think the power is, the openness of sharing the data across companies that make us move our products faster, our programs faster.
Great. Yeah. So I'm going to collate some of the questions together. There are several questions that really pertain to neutralizing antibody positivity before dosing. I just want to point out this is not the problem here. So the problem has nothing to do with the capsid, nothing to do with neutralizing antibodies. And so I will defer those questions for another time when we maybe can get together and talk about what it means to be seropositive and for future treatments of that. So apologies, but I'm going to move that for the interest of time to as well. There's a number of relevant questions that were asked is, now what happens if one or many sponsors get approval based on the current restrictions of the genotypes, and what happens to the patients that are included now after approval of this for clinical use, or will it not be approved with the exclusions? Just maybe one or two of the sponsors can give some thoughts on that. Carl, you want to talk about
that? Sure. I haven't sort of thought about it completely. I think as Teji said, the goal is to get an approved product out there. And we have to, unfortunately, this is an experiment. These are all experiments. And we have to sort of make sure that we have stringent criteria for those experiments. And so getting it out there and then making the argument to the FDA, and the expanded population, I think is sort of a more appropriate way of going is sort of if we're limited and then going in and there may have to be studies, trying to tolerize, immunosuppressant, other aspects that we talked about a little bit here. As we learn more, we might be able to open that up. But I think that the conservative approach, and I know it's not what everyone wants to hear, is to sort of really focus in on doing what we can to show that the microdystrophins are providing benefit to the boys, and then as we sort of move forward, we can really sort of try and get rid of the seropositive patients, so the exclusion around seropositive, the transgene and the mutations specifically.
So maybe I can take a slightly different one, and this will be really for you guys. Who is, and I think it's important for people to understand how things happen. Who is responsible for communicating findings and recommendation to the FDA? Well, to some extent, the FDA will give the recommendation, but anyway, communicating findings and recommendation to FDA and the Duchenne community. How does this work? I suppose, for the FDA, I think there are legal obligations that your companies will have. Maybe you can briefly describe what these are. And also, what are the steps? I mean, the steps today is one of the steps, but independent from these steps, what are the steps that a company do to keep the patient community informed of what's happening? One of you need to start, and I can see that, Serge has remained very quiet so far. So Serge, do you want to just very briefly say what are the regulatory obligation of the company, but also I think at your company or with the company where you collaborate with has, for example, issued a press release that was circulated to the various advocacy group and so on and so forth. But these are just examples. But do you want to elaborate further?
Yes, a little bit. So first, we, of course, provide the information immediately to the French regulator because the serious adverse event happened in our trial in France. And we hold the trial, and it was put on hold by both ourselves and the French agency. We let the patient group, the Duchenne patient group, know about the event, and we were lucky to have you as a principal investigator of this trial and have you, together with Carsten, committed in gathering the different sponsors. And it was very natural for us, as well as for the other sponsors, to meet and discuss the different cases. In our case, it was our first patient. And so we stopped the trial after injection of our first patient. And we went back to the regulator with more data and with the hypothesis you described here. And we will resume the trial in the coming weeks as the French regulator gave us the green light to inject more patients based on exclusion criteria, which is related to the mutation, and also with the an additional immunosuppressive regimen as an additional precaution to make sure nothing happened to the next patient. And Geneton is a non-for-profit organization, is a non-for-profit biotech held by AFM Teleton. So there is no question that we are fully transparent, and we want to make sure that the trial is done safely, as we learn from what is happening in our case and in the case of the other sponsors, and also to make sure that at the end of the day, this treatment will be made available for all patients in the future, including those that are at risk. But at this stage, it's very premature to say that patients that all patients can benefit from this gene therapy.
Thank you, Serge.
And maybe I could just add one piece onto that. You know, as Serge mentioned, each of the sponsors of a trial have a responsibility, let's say, to the regulatory agencies to report on things like SAEs, and you know, of course, we all do that. But I think what's really special about what we're doing here is that we took a sort of collective responsibility to not only share with each other, but to share with the community. And this is the unusual thing about what's happening here, because usually when there's SAEs and ongoing trials, you know, that's not necessarily something that's shared, you know, every moment with the community. But I think an important answer to the question is that we have taken this sort of collective responsibility and, you know, are sharing with the community, like in this webinar and in the presentation we're taking at MDA. And, you know, I think we all took that very seriously and will continue to take that seriously. So I think it's worth highlighting the unique aspect of this.
Yeah, that's true. There are a few questions also in the jumping over to the chat a little bit on mutations, specific mutations and their risk, and maybe we can just address some of those. So one is duplication. So at the moment we have no reason to assume that the duplications put patients at higher risk. They make because they don't necessarily lead to the not recognition of protein, to the loss of protein. So it hasn't been occurring in any duplication cases, fortunately. And then a question to very early exons, like exon 1 or 2 in the dystrophin gene. Also, they seem to be at lower risk. The reason for that is there is actually here in exon 5, there is a special site that allows protein to be made downstream of exon 5, even if upstream of exon 5 things are missing. So that allows the body to still make some protein to be visible to the immune system that is further down from exon 5 onwards. And that is good because what I haven't told you yet, it looks like the most critical region for the most immunogenic region, the most region that tickles the immune system the most is around here, starting with in that region here. And so that's the one that the immune system likes the least if it's not present, and seems to tickle the best immune response or the most vicious immune response. And then there's a question specific to some of the microdystrophins, let's say here in the solid microdystrophin or in the Pfizer microdystrophin, where there are bits and pieces of internal dystrophin in the microdystrophin. What about patients who have deletions in this region?
Yeah, I mean, I think that, again, to be conservative during this stage of development, that where we're considering that we've seen, you know, an early spot response to one of our patients in this region. And so as we sort of move forward and think about how we're going to sort of amend our protocol, this region definitely is under consideration, given that would be a new protein that's produced by our microdystrophin.
Yeah, and I'll just say from the Pfizer end, again, we've really been dependent on the data that we've seen so far in our studies and the patterns of mutation. So in the area of between exon 45 and 50 is a very common area for mutations. So we have a lot of participants who've been treated so far that have mutations in that area, and we have not seen any similar adverse events. And so just based on our experience, it doesn't look like that is an area for us that's at higher risk. Understanding exactly the biology behind all of that, I would say there's still some more to be learned, but we do have experience at this point that makes us think that those particular regions are not putting patients at higher risk.
And that's our assessment, but again, it's sort of, you know, given that we didn't see any clinical events, but we're just, you know, we may need to be cautious.
Mm-hmm. Excellent. I have maybe a slightly different question, but I think is the reverse of what we're discussing. Is there alternative work to produce microdystrophin that would not give immunity? And one of the questions in the chat was, what about full-length dystrophin? So in actual fact, the full-length dystrophin will probably continue to have the offensive antigen. So that is probably not the solution of this particular aspect. So perhaps this question could be expanded into part. Firstly, is there work that is ongoing to try to produce a non-immunogenic microdystrophin? But I think more in general, what are, again, without promising the things that we do not know, but what do you think are likely steps to allow in the future, when people are more comfortable, we understand better, to allow dosing also for individuals who have the at-risk deletions? So the first question is there work that has started to produce a less immunogenic dystrophin, number one, and secondly, would immunosuppression at some point be something that could be considered for some of the children with deletion in the at-risk area? I don't know who wants to take it.
Sure. I can start first and then have the others weigh in. So the short answer is, I think we're not yet starting work on a less immunogenic dystrophin. The reason for that is I think we're still trying to solve at Sarepta, and I think with the others as well, figuring out actually why this is happening. So I do want to be very clear that what we're presented here are hypotheses or possible reasons for this. These aren't yet fully defined conclusions. So I think our first set of work is trying to figure out, and we had a single case, but we had 8 other boys that had some mutations that didn't respond. So exactly what is the immunogenic reasoning for this? What is the hot spot? What puts somebody at risk, what doesn't? And then when we identify that, then I think that'll drive our next development. So I think our first focus in the short term really is helping define who is low risk, medium risk, high risk, what interventions are needed, and with the current construct, if we identify what is actually the trigger point, what can we potentially do to change the construct to allow that? But I think we're still in the information gathering, data collection, better understanding, because the number of cases are relatively low.
Yeah, and maybe I'll add to what Teji said, which I agree 100%, is that in this hypothesis that we have, there's nothing really wrong with the microdystrophins that we're producing. They're the same actual sequence as the full-length dystrophin, but just missing some pieces. The problem really comes in the disconnect between what the body has seen before from the endogenous dystrophin and then what the microdystrophin or the mini dystrophin is presenting to the body. So I think, you know, one way forward with patients who do seem to be more at risk because of their underlying mutation is to think about some immune medicines that can be given before dosing to try and sort of hold back that immune response. And, you know, I think that that's a reasonable path forward. You know, as Teji said, we have to understand it and we have to test those different medicines to see which ones work and how to best treat patients who have at-risk mutations. And I think that will come with time, and as we can sort of develop aspects of our studies to introduce those approaches. But there's not going to be sort of a quick fix. It's something that we'll have to move slowly and test out the best regimen to be sure that those patients can be dosed safely.
No. So if I can just add, and then Carsten will take the next one. I think I started by saying we know what we know, we don't know what we don't know, and this is happening now. So I think that is a much better step of it knowing nothing. But I think from there to say we understand everything, I think unfortunately there is still a space. And I suppose you can look at it from the other perspective and say it's very good that the majority of people, despite the fact that they may have large deletion, don't develop any problem. You can look at it from that perspective. That may not be the same for other conditions that are nothing to do with Duchenne. So let's not only concentrate on the half empty part of the glass. The great majority of the patient have not developed this problem. Let's not be, let's not forget about that. Carsten, you want to take the last one?
Yeah, I was just going to also end on a positive note. The more we drill into the actual mechanism of what is actually triggering the immune system and in what circumstances, the more we can react to it in a very targeted way, because there's a huge body of knowledge in immunology, in transplantation, in T-cell immunology. We're not starting from scratch. We just have to pick the right tools off the shelf. And the best way to do that is by understanding this in the proper way. The ultimate goal is tolerance. So we want to tolerize the body to this mini dystrophin, even if it may upset the immune system initially, doesn't mean it won't be tolerated later on if we do it right. What we want to avoid, though, is this kind of, someone put into the chat, muscle is a very immunologically active tissue. So muscle is very good at exposing things through the immune system and get attacked in response. And so the risk there, of course, is that the attack in response will get rid of the microdystrophin as well, or at least destroy some of it, and then the long-term effect is no longer assured. And so we need to make sure that we avoid this initial attack and induce long-term tolerance. And there are many, many different ways to do that, as long as we clearly understand what's doing that. And that's, I think we've made big strides in that understanding already, and things are cooking in the various labs to understand even more of it. All ensuring safety and tolerability.
Maybe I can add something to that, because we have some experience in another trial in a brain disorder, because there is little known about anti-transgene immune response in human gene therapy trials. We have treated 4 children with Sanfilippo B disease, and they had to be put under immunosuppression, prophylactic immunosuppression based on animal studies. And especially one of the 4 patients has improved because he she actually, a girl, was treated very early on. And she's now off immunosuppression and still doing well. So in theory, it could be possible to reduce and taper immunosuppression and up to no immunosuppression anymore. But in Duchenne dystrophy, we need to be even more cautious because exactly of the point you made, Carsten, that muscle and even Duchenne muscle is very prone for inflammation and potentially immune response. And we have to move with a lot of precaution. It might be possible to overcome the anti-transgene immune response in our trials, but this will take time.
Mm-hmm. Yeah, and maybe to acknowledge also what's happening in the chat, there is talk in the chat about inducing tolerance before the gene therapy. That, of course, is a very valid strategy and is being explored in a number of circumstances. It's not impossible to tolerize the body to peptides and things of that nature. That's been explored in other gene therapies as well, with targeted expression in the liver, for instance. There are many different strategies one could take. And of course, the very, very young immune system may be more tolerogenic than a later immune system. So all of these are very good points that are in the chat, and they're on the table as well.
Carsten, how long is this call? I'm happy to stay on the call. We've been an hour, isn't it? On the call?
We were slotted for an hour.
And so I'm just worried that in my institute, what will happen if you book it for an hour? After an hour, everything shuts down. So
we have Christoph in control, so we're safe here. But of course, we want to respect everyone's time. I do feel that we have addressed the big items that were coming up. There were a few questions that were sponsor specific that we got pre-mailed. Maybe what we'll do is we'll distribute them to the sponsors that got the question, and maybe you can respond then via Christopher to those specifically. I think if anyone has a burning question that wasn't really addressed, now's the time to just speak up. Otherwise, we'll be nearing a close. Oh, there's a hand up here. So the final hand goes to 076402, whoever that is. Can you introduce yourself, please?
Sorry, I didn't mean to be under the pseudonym. My name is Darek Gurecki. I'm professor of molecular medicine at the University of Portsmouth. My question is really about the immunogenicity that you've noticed. Is it possible that this is just the tip of an iceberg, that you see this rampant immune response in those 5 patients, but actually there will be immune response and therefore the rejection of all the fibers expressing mini-dystrophin, perhaps slower, but well, inevitable. Yep. If COVID vaccination taught us anything, is that intramuscular delivery of a potentially immunogenic protein results in a very effective and protracted immune response, both cellular and humoral. And of course, T cells, cytotoxic T cells, will eliminate all the myofibers that express mini-dystrophin. So hence there was a subsequent discussion about tolerance induction, the kynurenine pathways, exploration, perinatal tolerance induction, but sort of giving small dose of the vector to babies in the neonatal period. So I think it's just basically a bigger problem, and you may be only looking at the tip of it currently. Thank you.
I think those are valid considerations, and I think everyone will monitor very carefully for subclinical immune responses, influence on tolerability and longevity of the transgene, elimination of the transgene, and all of that. And then there's probably, I would think there's probably a nuanced answer to this, where we really have these top immunogenic epitopes that are really triggering this vicious response versus ones that are flying lower, and those certainly may exist, and one has to figure out what they do to the transgene and the durability of the expression and whether they deserve intervention as well. Those are very good points, and I think everyone is now monitoring very carefully for that.
Yeah, as it is important to finish on a positive note. I think that this is a valid point, and again, we know what we know, we don't know what we don't know. However, the data that have been released by some of the sponsors, I don't even remember which one, to be entirely honest, of comparing biopsy taken at different time point that will eventually be important to see whether this is going down, is going stable, and so on, have, if anything, indicated that the second biopsy had a little more dystrophin. And I think I don't remember the detail, but certainly not less dystrophin than the first one. So I think eventually the proof of the pudding is with time, with this confirmed. But I think that the evidence so far suggests that this is not happening, at least in this relatively limited number of children who had this biopsy. I think we're not talking about 5,000 children, but I think this is I remember having seen. Every time I see those data, my heart gets a little warmer.
Yeah, and I guess I would add to that. I totally agree with you. And in a lot of the studies that have been ongoing, they've been ongoing now for years. And so we've had a chance to observe the boys' performance, their activity, their biopsies over a very long period, much beyond this sort of 1 month time period when it seemed like there was an immune response. So of course there's more to come. These are all ongoing investigations, but so far it doesn't look like there's sort of some other smoldering low-level immune responses that maybe didn't reach this level, but that are clinically important. It looks like really that it is targeted to a specific set of at-risk patients.
Great. On that note,
so I think that I would just say one thing, and then you conclude. Because I think that well, some of you ask what are the next steps. To some extent, I think Carsten and the other colleagues have indicated. I apologize. I need to drink something. I promise it's not COVID. I just had my test because I'm flying to Amsterdam tomorrow, possibly. And we do not necessarily intend this to be like a recurrent meeting. At the same time, if you have specific questions that are not being addressed at all, send them to us. We'll do our best to look for them. I think that there may be, if there is the demand, if there is new knowledge, we could discuss. We are not committing anything because I think again, we are not sure, but there may be a possibility of other events for sure. This group will ensure that whatever will be written, Carsten mentioned some different type of papers will be distributed with all of you. And I just thank the companies, but Carsten in particular, and Mike, over to you.
Well, I think I speak for everyone to thank all of you to join us today, and I hope we were able to answer some of the questions, some of the concerns, and also point to a way forward. I think that the goal of everyone in this room is to be able to treat every single boy with Duchenne muscular dystrophy, but we have to do it in a safe and effective way that doesn't put patients at risk or programs at risk for that matter. I still want to emphasize that I think this collaboration, this coming together of the sponsors around a class effect, a class toxicity, if you will, something that the entire field needs to address, is precedent setting, and I hope it'll become standard in our field to really address problems like this as we do today. I'd like to congratulate everyone involved in doing that. Any final words from you, Beth? Because you put in so much work in this as well.
No, it's been a really wonderful experience, and thing for all of us, and we enjoy working together, and yeah, and we'll just keep going.
Thank you. We hope to continue this at some point. Thank you. Good evening, or wherever you are.
Thank you, everyone.
Bye-bye.
Bye.
Good night. Bye-bye.